Patty, why don't
So,
you start?
we welcome. Everyone is here will be able to join in. Tina will keep track for us. Good evening and welcome everyone who has chosen to join us today. I am Patricia Cannon, Chair of the ACKCS Charitable Trust. Thank you all for joining us this evening. And most of all, thank you for supporting us in more of the groundbreaking work that we do and our planning for the future. I would like to introduce Miss Tina Sterling, who is chair of our Science Advisory Committee and is also, goodness, I'm very sorry, that's my fault, a medical health administrator, OB/GYN nurse. She has tremendous experience in her professional life in the world of human and animal reproduction. So here we go, Tina.
Well, I'm going to say hello and welcome to everybody. Tonight's seminar, as you know, is Breeding for Heart Health and a Holistic Approach with Claire Wade. I would like to thank the ACKCSC Charitable Trust for hosting this webinar and the CKCSC USA for supporting it. There's always housekeeping, so here we go. There will be a Q&A session at the end of the presentation, but the chat will be on continuously throughout the excuse me, the webinar. And Dr. Wade said that she would like people, if she's talking about something that you don't understand, put something in the chat. Oh, the chat is disabled?
Yeah.
Okay, I'm sorry. See, not as,
Housekeeping.
yeah, I'm not as that good with.
Just keep going. I'll see if I can fix it.
Okay, my daughter's going to see if she can fix the chat. Anyhow, the Q&A is going to be at the end, and we were going to it's 30 minutes or longer depending on how many questions we have. And if we don't get all the questions answered, then I will send them to Dr. Wade, who can answer them, send them back to me, and we'll put them on the website. I would like to ask and encourage everyone to go up on our website. The website is being revised a little bit at this time, but it will include the results of surveys, projects, testing, updates on grants funded, and current scientific information. I'd like to mention our new health survey that has just been launched this past Tuesday. It is a joint venture with our trust and the CKCSC USA. Unlike many surveys to date, this survey will be overseen and run by Dr. Jason Stahl, a veterinarian and epidemiologist from the University of Prince Edward Island in Canada. Dr. Stahl is also very well known. He is a keynote speaker this year at the CHF conference in August, where he will be awarded the prestigious Asa Mays Award of Excellence. Just want to mention the survey is anonymous. Dr. Wade also has given us permission to record her presentation, and we will be adding it to our YouTube channel that you can find at ackcsccharitabletrust.org. Okay, enough of that. I'd like to introduce Leslie Slusher, who is a retired professor of genetics and biology at Westchester University. She serves on both boards, the ACKCSC Charitable Trust and the CKCSC Scientific Advisory Board. She will be working on statistics and data from the results of testing and the survey. Professor Kathy Yonkers is a subject matter expert in research and health economics. She's a retired public health practitioner, bioterrorism coordinator, and epidemiologist. Kathy is currently serving on the ACKCS Charitable Trust Scientific Advisory Board. Kathy is responsible for asking Dr. Wade to present and organizing tonight's webinar. Okay, I know that was a lot and a large introduction, but now, Kathy, can you please introduce our presenter, Dr. Claire Wade?
Well, I'm very pleased to share with the audience that our speaker tonight is, or whatever time zone you're in, is Professor Claire Wade at the University of Sydney. She is a pioneer in genetics and I would like to have her share her vitae. And the presentation will probably go to about an hour. So she has a lot to share, and I'm looking forward to it. So, Dr. Wade, would you please share your background?
Oh, okay. Stop talking. Okay, so sorry. I'm Australian originally. I lived in America for 7 years. I'll talk about that in a moment. I studied at the University of New South Wales in Sydney, Australia, where I studied animal science before doing a PhD in livestock selective animal breeding. When I finished my PhD, I worked for a while as a sheep geneticist with the Victorian Department of Agriculture in a different state. And then I moved to the University of Queensland, where I was a junior professor teaching genetics to veterinary science students in the Faculty of Veterinary Science. And I worked there for 10 years teaching veterinary students. And that took us up to about the time that the human genome was first released. And I guess for me, up until that time, I had been trained in classical animal breeding, selective animal breeding, and I felt as though I had kind of missed the boat with the gene mapping part of the world, even though that up until that stage, gene mapping had not been particularly successful, I might say. And so I had a sabbatical due to me through the university, and I wrote to the people who had sequenced the human genome and said, can I come and work with you? And they said, yes, please come. We really need people like you who have your skills, because I was a geneticist and also I could do computer programming. I have computational skills. And so I went and worked at the Whitehead Institute at that time with MIT, working on the mouse genome project. And at the conclusion of the mouse genome project, the next mammal that we were interested in was the dog. And I had always been interested in dogs and horses. And actually, the breed that I have now, I have Nova Scotia duck-tolling retrievers, and I fell in love with those while I was actually collecting samples for the dog genome project while I lived in Boston. Anyway, the dog genome came out, and I played a large role in that, doing the population genetics and doing all of the variant discovery, and also creating a number of genetic resources for dogs that are still out there now, including the reference genome and several what we call genotyping arrays, which are sets of markers that we can assay across the genome for the dog to help us to do gene mapping. And then after that, I worked on a few other mammal projects, but then finally at the Broad Institute, the Broad Institute, which was a collaboration between MIT and Harvard, came about, and I became a member of the Broad Institute, and I took over the leadership of the Horse Genome Project. And at the conclusion of the Horse Genome Project, I returned to Australia because I felt as though well, first of all, I was homesick. My parents were elderly, and I felt as though it was time to do something a little bit different. So I came back to Australia and took my current role, which is as a professor. At the time, I was the first female professor in the Faculty of Veterinary Science at the University of Sydney. But since then, I've actually been moved to a different, the Faculty of Veterinary Science doesn't exist anymore, and I've been moved into the Faculty of Science in a different school called the School of Life and Environmental Sciences. But really, my first love is I love working with genomics, and I love working with genomics to improve canine health and welfare. And most of the research that I've done over the past 15 years or so has been dog-related, working on all sorts of things, creating genetic tests. I'm actually an inventor on the degenerative myelopathy test, which is so controversial. But I'm an inventor on that test, and I've also created several other genetic tests that are used commercially out in the world. In fact, the Neville test, I did not create that one. We just validated it. So that's a brief history of what I've been up to. I have lots of publications. Many of them are dog-related. I have a few other species thrown in there. But hopefully my background in both selective animal breeding and genomics and gene mapping can help me to give you some insights that you might not have otherwise had. And so that's what I mean by holistic approach. We're not going to be completely gene jockeys today. We're going to consider other things as well. So, yeah, so that's me in a nutshell, I
Thank
guess.
you. Thank you
Oh,
for
I
sharing.
didn't say also. I breed Nova Scotia duck tolling retrievers. I'm actively involved in lots of canine performance sports, and I'm actually a retrieving trial judge. So I do lots of things with my dogs. My dogs always have titles at both ends and lots of versatility awards and so on. So that's
Wonderful.
my goal, is to produce an all-round dog that fits the breed standard and can do whatever the breed standard suggested it should do. So, yeah, sorry. I thought I'd put that in there as well for the dog
Yeah.
people.
Wonderful. Thank you. Okay.
Do we want to launch the presentation?
Yes,
Sure.
I'm sorry, I was on mute. Yes, please launch your presentation.
Okay, so we're going to be talking this morning about general breeding for heart health, not only in Cavaliers, but they're going to be the focus of what my talk is related to this morning. We're going to cover, let me see if I can advance my slide. Ooh, okay, that's what I didn't practice. It's slide advancing. My mouse is, oh, here we go. Okay, so sorry, I had to tilt my screen so I could see the next thing. So this morning we're going to just cover myxomatosis mitral valve disease, because this is a major heart health issue in the Cavaliers. And then I'm going to tell you how this Neville gene story came about, and what was the background in that, and then where we became involved. And then we'll talk about how you can different ways that you might include that information in your breeding program. Then we also have some new research. Well, it's new to you. It's not really new to me, but we have. I had a PhD student working on this for some years, and we have some research that's yet to be published. That's currently trying to put the publication together now. That might present some more opportunities for improving heart health in the Cavaliers. And then we'll talk about some other selective approaches that might help heart health that are not necessarily related to gene testing. The next thing I'd like to just do is if I have a problem that my slide advancing button is on the other screen and I can't see it when my screen's up. Okay, so I just thought I'd cover some basic terminology, things that I say often that I don't even think about, but you may not understand what I'm talking about when I mean it, when I say these things. So when I talk about genetics, really genetics encompasses all these things, but when I'm talking about genetics, I'm talking about selection of animals, but not necessarily using genetic testing. So not usually selecting animals based on things that you can see or that you have measured at the cardiologist or at the veterinarian. Those things I regard as genetics. Genomics I regard as things that actually are involved with the operation of the molecule of DNA in the cells. So it's more of a laboratory-based, DNA-based testing that we use for selecting breeding animals these days. Another word that I might use is phenotype. So when I talk about phenotype, what I'm talking about is a physical trait that you can either measure or assess visually or assess in some other way. That's not a laboratory test usually. If I talk about an assay, that's usually a laboratory process that's used to read a gene or a variant, and I'm going to describe those things in a moment. And I don't know how familiar my audience is with basic genetics, but you may know that genes come in pairs, and that when the new embryo is formed, it's formed by components that come from both parents, and the genes come from both parents. So our genes are in pairs, and one of each pair comes from mum, and one of each pair comes from dad. And so the genotype is that combination of the two genes together. that that we might be interested in. And so when we're talking about a carrier, we're talking about an animal that has a different gene from mum than what it had from dad. And the genotypes, for things like coat colours, sometimes we can tell what they are by looking at the animal, but oftentimes they have to be assayed by a genetic test. And then a gene, so the word gene is a little bit changeable these days, because in the old days it used to just be a segment of DNA that coded for a protein. But I think when most people use the word gene in their everyday speech, what they're thinking about is a DNA-based element that affects the phenotype, what we can see and what we can measure in the dog. And so it's something that has a functional outcome in the animal. And lastly, a variant is a small change in the DNA. So the gene that you get from mum and the gene that you get from dad may not be exactly the same. They might have variants in them. And variants, these little changes in the DNA that make us all individuals. And every individual has their own DNA that's just theirs and nobody else's. Even if you're an identical twin, very likely there are some little pieces of the DNA that are not quite the same. But the ones that we actually use to do gene mapping or to use a genetic test, we call that a variant, because that we know that there's a change there and we can use it to provide us with some information. So that's just some basic terminology before we get started. And also I want to bring to your attention some really, really valued colleagues that I could not have done this work without. And first and foremost, Dr. Nick Beierink, who's a cardiologist, works in the Netherlands. He was at the University of Sydney and comes back every now and again. He's still affiliated with us, but his major place of work is in the Netherlands. Mitchell O'Brien did his PhD on our Cavalier King Charles project, and he actually was responsible for doing the mapping that I'll discuss later in the presentation. And Sophie Mead is, at the time that we did the Nebel work, she was an undergraduate student working on a research project for her undergraduate degree in animal veterinary biosciences. But she has since graduated and is now a Doctor of Veterinary Medicine candidate at the University of Sydney. And she is currently working with me on a different project relating to primary ciliary dyskinesia in Cocker Spaniels. So Sophie was the main architect of the Nebel work that we did, and a very valued member of our team. Sorry, I'm going to find my spot again. All right, so most of the work that we do in our lab revolves around measurements of heart size that are assessed by echocardiography. And so I thought I'd just outline for you what the measurements are that we're looking at. So here's the canine heart, and the measurements that we are interested in relate to the size of the heart, and they're trying to adjust it for the body size of the animal, because a bigger animal will generally have a bigger heart. But we need to know if the heart is bigger than it should be, because when it has to work harder because it's not working effectively, it's like every other muscle in our body. If we lift weights or anything, our muscles get bigger. And so if your heart is lifting more weight than it needs to, the muscle starts to grow, and that's a sign that the heart is not working effectively. And so the echocardiographer will take certain measurements that they can use to assess whether that heart is working harder than it should be. And the two main sets of measurements that are used are, first of all, the ratio of that red arrow to the yellow arrow, and that's the left atrium. So the atrium being the upper chamber in the heart, the diameter of the left atrium compared with the diameter of the aortic root. So you might remember that the aorta is one of the major blood vessels in the heart that carries the blood from the heart. And the aortic root is thought to be fairly proportionate to the body size of the dog. And so if the left atrium is larger than the aortic root, that tells us, or is larger than expected for the aortic root, then that tells us that there's a heart size issue happening. The other measurement that's used is the sorry, just get my mouse right. The left, so the ventricle is the lower chamber in the heart, and the left ventricular diameter is another measure of the heart. So it's measuring the lower chamber this time, and this measurement has to be corrected for the body weight of the dog. So these are the two measurements that the cardiologist uses to ascertain whether your dog's heart is bigger than it should be. So the cardiologist, I have to, as I've pointed out, I'm not a veterinarian, so you need to forgive me if I make fluffs on the veterinary terminology. I was relying on Nick to do all the veterinary work in relation to our projects, but as I'm sure that most of you who breed Cavaliers are aware, they have different staging of disease, and in other breeds, anything beyond stage A is regarded as subject to heart enlargement, and stage A is the healthy level. In Cavaliers, most Cavaliers will proceed to stage B1 at some point in their life. In Cavaliers, usually the cardiologists are concerned when you get beyond stage B1 into B2, C, and D, and then eventually at the higher stages, it will end up in a state of heart failure and the dog might pass. So there are certain measurements of those two measurements that I described earlier. There are those particular ratios denote the boundaries of what's considered healthy and what isn't. And the stage B2 is where that boundary is put into place for the Cavaliers with respect to them starting to experience heart enlargement that might result in heart failure. Okay, so so there is mitral valve disease. It is true that there is some mitral valve regurgitation in most Cavaliers at some point during their life, probably more than 90%. And that mitral valve disease can lead to heart enlargement and heart failure. But not all dogs suffer clinical symptoms related to that, and so there's no impact on their quality of life. But some of them really do, but the trouble is that when they're young, it's really hard to know which dogs are going to go in which direction. And so ideally, you can see from that chart, what we're looking at on the x-axis is the age of the dog, and on the y-axis is the heart size measurement. And usually the dogs on the blue line, they'll just truck along and they'll be happy all their life. But the dogs that take the green path, usually they proceed very rapidly to heart failure and are lost. And so ideally as breeders, what we want to be able to do is to distinguish those two groups while the dogs are young. But you can see that in the early part, up to 6 years of age, it's very, very difficult to tell those two groups apart. And so we're looking for solutions that can help breeders identify those two groups of dogs earlier in life. And genetics can help us to do that. Okay, so it's widely known that there is quite a lot of MMVD in Cavaliers, and as a result of that, a group from the University of Uppsala in Sweden, and just as an aside, Kirsten Lindblad Toh, who's the final author on that list, was the person that I worked with while I was my primary person that I worked with at the Broad Institute, her and a guy called Mark Daly, who's a very well-known human geneticist. And Kirsten is an extremely accomplished, brilliant scientist, and she's very considered in her approach to science, and I do trust the work that she produces. In this study, they had not initially set out to just look at Cavalier King Charles Spaniels. They had actually set out to look at a range of different breeds. But they were sort of interested in the problems that were regarded as primarily afflicting certain of the breeds and that had been resistant to selection. And so this study was looking at possible fixation for genes based on the analysis of the DNA for a few different breeds. And when they did look at the Cavaliers, they did find some heart genes that appeared to be fixed, by which I mean that any gene you get from mum and any gene that you get from dad are going to be the same or close to fixed in the Cavalier King Charles Spaniels. And that was sort of the main result they had. And so as a result, the paper, when it was written, focused very heavily on the Cavalier King Charles Spaniel outcomes and kind of didn't really spend so much time discussing the other breeds that they looked at. So actually in their study, they had 1, 2, 3, 4, 5, 6, 7, 8 different breeds. So they had Beagles, they had Cavaliers, German Shepherds, Golden Retrievers, Labrador Retrievers, Standard Poodles, Rottweilers, and West Highland White Terriers. Now, first of all, there were only 20 individuals, but I know it sounds like a small number, but this is a big deal, right? Because you have to remember that they're reading every letter of the DNA. They're not just having a look here and there. They're looking at every letter of DNA. And if you think back to when the human genome was sequenced, which is the time that I became involved in genomics, it used to cost millions of dollars to sequence an individual genome. And nowadays, we're lucky it costs less than $1,000 to sequence a genome. But it's still, even at $1,000 a genome, this is still quite an expensive project. If you're doing 20 times 8 times 1,000, that's still a lot of money. And I have to tell you that in dog research, money is not very available. Becoming less so, because the NIH is shutting its doors down a little bit. All right, so they sequenced the whole genomes of these different animals. They read every letter of the DNA, and dogs have lots of chromosomes compared with most animals, and I think this is part of the reason why it's so easy to map genes in dogs. They have 38 pairs of chromosomes plus their sex chromosomes, so plus an X and a Y if you're a boy or 2 X's if you're a girl. So what they did was they detected all of the variants in those genomes, and they assessed whether they occurred in places in the DNA that might do something to affect the consequences for the animal in a meaningful way. Because there are lots of mutations that occur that do absolutely nothing. The vast, vast majority of variants in the genome do nothing at all. They just act as signposts for us to help find things. They don't actually have any function. And so what this group did was then they looked at those different variants or mutations, and they identified the ones that they thought were most likely to have a deleterious or bad effect on the health of the animal. And so as part of that, they, for the Cavaliers, we'll go to them, they looked for, they had a list of genes that they thought had a good possibility of being involved in heart function, and they looked at those genes with particular attention, and they identified a set of different variants that appeared to be fixed in the 20 Cavalier King Charles Spaniels that they had sequenced. So in those 20 dogs, all of them were the same, and they all had a variant or mutation that was different than the normal one, and that the scientists had assessed was likely to create some damage in the physicality of that animal. So the other thing that they filtered on was that for those variants that seemed to be fixed in the Cavaliers, they were looking for ones where all of the other dog breeds had the normal version. So that not only were these variants very high frequency in the Cavaliers, but they were kind of out on their own as having these variants that were largely fixed and probably creating some physical change. And so they created this set of genes, these variants like this, and then they started to look at that reduced set in more detail. And so the scientists were thinking that these specific changes in the Cavaliers might be responsible for the specific physical problems that were observed in the Cavaliers. So at this stage, they had quite a few mutations to work with, and they had to filter them down. And at the end of the process of their filtering, they had various other filtering methods that I didn't explain that are kind of technical, but they had good reasons for doing them. And at the end of this filtering process, they identified 10 mutations that were most likely to be heart health related deleterious variants and fixed in the 20 Cavaliers that were tested. And it happened that of these 10, 6 were near the gene called Nebel. Nebulin is the proper name for the gene. So there were 6 variants in that region. Now, as a geneticist, when we're gene mapping, one of the things we look for is you don't want to signal, if you're gene mapping for something that's important, you don't want something to just be by itself, because that usually is a result of some technical error. So the fact that there were these 6 variants that were behaving the same way gave the researchers confidence that this gene really did have something to do with what they were seeing. So now, because the 20 Cavaliers they had were all the same for this variant, the researchers had proposed that there would be no variation in the Cavaliers, and so they couldn't test these variants for function because there was nothing to compare them to. So you want to compare apples with apples. So ideally you want to compare Cavalier heart health without the gene to Cavalier heart health with the gene. And they weren't able to do that because all of their Cavaliers that they tested were all had the bad version. And so what they did was they took it to a different breed, which was the Dachshund. And the Dachshunds have this variant as well, but not fixed. And so they were able to identify 122 Dachshunds that had echo results and had also been assessed for mitral valve disease. And they were then able to compare if these different variants had an effect on the apparent staging of heart disease in this different breed that had the same gene variants segregating. And so the end result was that they had 122 dachshunds that were tested at the Swedish University for Agricultural Sciences in Uppsala. 79 of the dachshunds had MMVD, and they were counting the B1s in the dachshunds as being in their case set. And then they had very few, they had 43 dachshunds that were considered healthy with the staging of A. And so they were able to compare the gene variant risk for the Nebel variants in these two groups of dogs, and they saw a difference. That made it look as though the Nebel 3 variant and the Nebel 3 and Nebel 4 variants appeared to be the ones that had the biggest effect. And then they followed up with some fancy laboratory stuff that supported this finding. So when we map a gene, usually you don't really believe the mapping unless you see it a second time. And so what the Swedes were doing was that they were testing this to see it a second time. We call that a validation. So in their mind, they had validated this change by looking in the Dachshund breed. But it hadn't been, so this is kind of where we start to come in, because we found study. And we had a lot of Cavaliers that had echo, and we also had DNA from those Cavaliers because we were already using it for something else. And so at this stage, Sophie came along, and we thought, well, it's all very well to validate it in the Dachshunds, but even a better validation would be to see if it does something in the Cavaliers. But the first question we had was, is this gene really fixed in the Cavaliers? Because the Norway breed bans had come along at this time and they'd said, all of the Cavaliers are unhealthy, therefore the breed should be, banned, blah, blah, blah. And so our question was, well, there really were only 20 dogs in this primary analysis. Is that the whole story? And so we wanted to look and see if there was some possibility of there being good genes in that population. And so Nick had been doing heart screening, Dr. Bayerink had been doing the heart screening for Australian Cavalier breeders for some time and collecting DNA, and Mitchell had the project for mapping age of onset or staging for Cavaliers from a gene mapping perspective, which I'll talk about in a moment. Anyway, so we had close to 200, so that's quite a lot more than 20. And even though it's a very small number, among that 200 of dogs that were being actively screened for heart health, so it's probably the more proactive end of the breeder population. We've got Cavaliers being actively screened for heart health because the breeders care. They may have been inadvertently selecting for these good genes if they were there. And sure enough, we found 6 dogs that had the healthy version of the gene, but they were only carriers for that. And so Sophie came along and we looked at the same 10 variants that the Swedes had and the same 5 genes, because those 10 variants were in 5 different genes. And we looked in our data at how variable were our 183 Cavaliers at those different locations. And what we found were that 3 of the genes that the Swedes had been concerned about were clearly not fixed or even close to fixed in the Australian dogs. So we didn't have to worry about those. But there were 2 genes that behaved the same way in the Australian dogs as they had in the Swedish dogs. And those genes were Nebel and HDGFL1. So, and you might wonder why we've not been talking about HDGFL1, or will be revealed shortly. But crucially, and happily, we did find some Cavaliers that had the healthy versions of the genes. And because we had cardiologist assessment for those dogs, we could compare those healthy dogs that had at least one healthy version of the Nebel gene or the HDGFL1 gene with the rest of the dogs. And we observed, it shouldn't say we observed whether their heart size, we observed their heart size. And we found that when we looked at the heart sizes, the dogs that had one healthy Nebel variant had smaller hearts than the dogs that did not have that. But the same was not true for the HDGFL1 gene. So we observed a significant difference between at the Nebel gene in the health, the heart health of our local dogs, and we had considerably more dogs than the Swedes did, for the Nebel gene, but not the HDGFL1 gene. And that's why our paper concentrated on outcomes for the Nebel gene. And so I won't draw it so much to your attention, but I just really want you to, in the mean column there, so what we're looking at here are the different variants. Now, in our population, the Australian population may not be the same as the population elsewhere in the world because we are an isolated continent and dogs have to be imported here. So it's possible that things are not quite the same here as they are elsewhere. But in our population of the dogs, every dog that had the Nebel 3 normal variant also had Nebel 1 and Nebel 2 normal variants. But remember back that the Swedish dogs, they cared about Nebel 3 and Nebel 4. Anyway, so now, just a reminder that for our heart ratios, we have certain ratios that determine whether they're healthy or not. And that determines whether they're A or B1 versus B2, B3, C and D. Sorry, B2, C and D. And that ratio, that critical ratio is 1.6 for LAAO and 1.7 for LVIDDN. So what we're looking for are numbers in that mean column that where the dogs that have that variant are in the healthy range. And you can see that the Nebel variants are all inside the healthy range, but the HDGFL1 dogs were not. And so we concluded that the Nebel variant did truly appear to be a predictor of heart size, and that having at least one copy of that gene promoted a healthy heart size. And these dogs were, the values were corrected to an age of 8 years. So, yeah, there was some statistics behind it that led a little bit more weight to this. All right, so, sorry, my other dog's barking. All right, so remembering that the LAAO is the ratio of the upper heart, and the LVIDDN is the diameter of the lower heart corrected for body weight. Anyway, so our summary so far, the Nebel story. The Swedish group identified 10 gene variants that were in 5 different genes that were thought to possibly contribute to heart disease in the Cavaliers and appeared to be fixed. But they tested only a modest number of Cavaliers, reading every DNA. So I don't want to disrespect what they did because it was a big study, let me just say. They then tested the same variants in other genes, in other breeds, and determined that the Nebel gene locus might play a significant role in heart health. And then, so our group took all 10 of those variants and asked, are they fixed in our Cavaliers? And the answer was that only Nebel and HDGFL1 appeared to be high frequency alternate in the Australian population. So we kind of disregarded the other 3 genes. And then we looked at those genes to say, do they appear to have an impact on MMVD staging in Australian Cavaliers, and in 180 of them that had cardiology assessment. And we found that the Nebel gene did have an effect, but the HDGFL1 gene didn't. And so this is why it's all Nebel all the time. We're not talking about the other ones at all. But the other important thing we found were that some Cavaliers had the healthy version of the gene, so it was not entirely fixed. And so I guess our conclusions in our paper were, by far the quickest way to fix it, well, fix in the other sense, so to correct this problem would be to use outcrossing. But those of us who are pedigreed dog breeders all know that inside the kennel registry systems, if you want to stay in that kennel registry system, the opportunities for outcrossing are very limited. So we were interested to know if there were resources in the purebred population that could be leveraged to rapidly improve heart health in this breed, making use of this information. So I guess the other thing I just want to, this is a bit kind of technical, but there's other work, recent work, 2025 work, that shows that this Nebel gene is a risk factor for cardiomyopathy in humans as well. So there's quite a lot of evidence to say that this gene really does something with the heart. Okay, so then we come down to the breeder, more the practical side. So that's the backstory. That's where we get up to now. That's where we are in the world in relation to our information. But how do we effectively use that? And there are big concerns that breeders have in relation to this. And those concerns need to be thought about we need to consider them in the context of a breeding program that this is not the only problem that you're dealing with. So all of us who breed know that there are many things that concern us when we're breeding. Yes, there are many things. Every breeder's list is likely a little bit different, but there are many, many things that we are juggling when we're trying to juggle our breeding program. And we can't, usually, a lot of researchers who are not dog people, they think you should just go hard on this one thing and they'll fix everything. Well, we all know that's not true, right? So the important points are that heart disease is not caused by one gene, and therefore Nebel will never be a magic bullet. But it's never going to do away with MMVD in the Cavaliers, probably, even if you had everybody wild type. But what it might do is it might delay the onset of the disease progression to congestive heart failure to an age where it really doesn't matter so much. But when we want to include this gene in our breeding program, if you decide you do want to include some selection for this, then the other concerns you might have are that all of the dogs that have this wild type gene are nothing like the dog that you want to breed. And so this is a big concern for you. And the other problem might be, well, how do I find dogs that have these good genes? How am I ever going to know if they have them or not? And so those are things that we'll just discuss now. So, how do you bring healthy Nebel genes into your breed without losing breed type? And so it's well known in livestock breeding that we can use this system of back crossing and to do this kind of thing. And the Dalmatian breeders have been using this to bring in the hyperuricasuria healthy gene to the Dalmatian breed. In their case, they had to outcross to a different breed. In your case, you might be able to identify a purebred Cavalier that has the right gene, because there aren't many of them, but there are some. But that Cavalier may not be the breed type that you find to be desirable for your breeding program. And so should you be concerned about that? So what I would say to you is that it doesn't matter how different or unusual. Can I just give me a moment? I've got to, my girl needs to go out. Just a sec. Out you go, quick. Sorry, she was pounding on the door. Mothers. Okay, so no matter how undesirable that other dog is in terms of looks, it with a little bit of effort, it does not take long to get back to something that you would never know the difference. And so, just a second. Al, can you let her out, please? Al, can you let her out, please? Sorry, for the people that joined late, I had a litter on Saturday, and they're in the office with me, and the mother dog really needed to go out badly. Anyway, so because puppies get one half of their genes from mum and one half of their genes from dad, when you cross in that desirable, undesirable, the desirable genetically but undesirable phenotypic dog into your breeding program, the puppies that result will have exactly 50% of your breeding and 50% of the wrong breeding. Now, if you then take a puppy from that litter and breed it back into your desirable type, ideally the dog that you use initially would be homozygous for Neville, but it might not be. If it's a carrier, only half of the puppies will have the good gene, so you'll need to do genetic testing at each stage to determine which puppies have the Neville desirable type before you select them into your breeding program. So at each stage you'll need to do, if that first one was a carrier, each stage you need to do a genetic test to see that you're keeping the one that has the good genes, that you haven't let them go out the window. So if you do a test, if you do testing that uses a genome-wide testing, for example, EMBARK, if they use, or even if you use parentage markers, it is possible to accelerate this process, because after the first cross, there's a bit of shuffling that occurs. And so even though the first cross is exactly 50%, the next time you do a back cross, it's only approximately 25% the wrong time. There'll be a normal distribution around that 25%. So some will be closer to 12% and some will be closer to 30%. And so you can use, there are strategies you can use to choose the ones that are most like your type. And so you can accelerate that. But even if you don't do that, even if you just test Nebel, you can predict that approximately 75% of the genes in the second cross will be your genes. And the third cross, it's so close to being yours that you will never know the difference. So you can bleed in the Nebel gene into your desirable type. You just have to be prepared for a couple of generations to have something that's maybe not as perfect as you would normally see in your breeding program. Okay, so so it's possible to use this back crossing system to bring the gene in and to keep the majority of the things that you care about. It's most effective. You must use the Nebel gene test to make sure that you're not letting the good genes slip away accidentally. And if you use a genome-wide test, you can accelerate the process. Now, the issue here is that in every case, the puppy that you're keeping is going to be a carrier. It's not going to be homozygous for the wild-type Nebel gene. So at some point, you have to breed together two of these animals that have had that good gene brought in order to generate a dog that's homozygous for the normal gene. Okay, so a little plug for the Cavalier King Charles Spaniel Nebel 3 Breeding Project Facebook group, because there are a group of dedicated breeders who are interested in increasing the frequency of this Nebel 3 wild type in the Cavalier population. And that's a great place to go to get some support if you're interested in following this process, because there are a lot of like-minded people who are trying to increase the frequency, and I would predict that there are more likely to be good Nebel genes in that group than there are elsewhere. So if you want to find another breeder to work with, that might be a good place to go. All right. So what can you do? Well, the more people that use genetic testing, the more dogs that are carrying the good Nebel gene will be discovered. If you are interested in maintaining your breed type, then you choose a dog carrying the healthy gene that's closest to your desired breed type. You'd probably do that anyway. But even if you don't panic. Like, even if there's nothing of your desired type close to you, you can see that within 3 generations, you can be back to your desired type quite readily. So don't panic. It will all work out well in the end. But the important thing is to genetically test for that Nebel 3 variant at each stage in the process. And you may already have them. If you've not been testing yet, you might discover that you've got healthy genes there and you didn't know it. All right. So from I was interested to know how many good genes are out in the world. And from the Cavalier King Charles Spaniel Nebel 3 Breeding Project Facebook group, someone reported that Labaclin had done some testing. I was unable to verify this. I was not able to find these statistics on the web, but they reported testing 500 Cavaliers. 4 dogs had the clear, normal, wild-type variant, GG. 70 dogs were carriers, and 426 had the risk gene, which is the A variant at that locus. The locus is the actual variant location. Okay, now I also put some feelers out locally to see what was happening in the Australian scene. And there's a local testing facility, Dog Breeding Science. And I know Dr. Castle quite well because she did her PhD in our department, so I know her pretty well. And I asked her, would she be willing to part with her outcomes for her testing that she'd done at Dog Breeding Science. And so I got these results hot off the press last night. And in her testing facility, she doesn't ask the people doing the testing to report breed, because she works a lot with commercial dog breeders as well. And so there are a lot of cavoodle breeders as well. And so she's doing cavoodles, cavaliers, dachshunds, because they're the other breed from the paper, and poodles. And what was surprising to me when I saw this last night was that there's actually 11 poodles that are homozygous for the risk variant. So your cavoodles are also a high risk mix for this heart problem caused by that might be negatively influenced by the Nebel gene. Anyway, so now she's tested about 2,000 dogs, and of those that she's tested, she was unable to determine whether they were Cavaliers or Cavoodles, but she had 44 that had identified as dachshunds that were carrying the risk variant, and she had a few poodles here and there that had all genotypes that were being tested. And the critical number is the number in the normal cell. And so there were 39 normals. Now, some of those might be cavoodles, of course, but she knows that at least 2 of them were Cavaliers that were intentionally bred from carrier to carrier breeding. So there do exist homozygous Cavalier King Charles Spaniels in the world. They have mainly been produced deliberately by the people in the aforementioned Facebook group, I would predict. So that's the place to go to find the genes, I would say. Alrighty, so that's Nebel. That's all we know about Nebel. I can't tell you any more about Nebel than that. All we know is the Swedes identified it. They validated it in dachshunds. We validated it in Cavaliers and found that Cavaliers are not fixed. And there are Cavalier breeders who are conscientiously trying to increase the frequency of this gene in the Cavalier population so that purebred Cavalier breeders can access this gene that might prevent progression to heart failure. Alrighty. Now, just very briefly, of course, this is not our only project in this space. So the main reason we were able to get in on the Nebel action was that we already had this other project going. And so Mitchell and Nick and I had been doing some gene mapping for those 2 ratios that we had mentioned earlier. So the LAAO and the LVIDDN measurements. And so when we're mapping, what we're doing, we don't care about whether the markers that we assess actually have function or not. We're not caring about this at this point. We're just looking for signal. And remember I said that we want multiple signals in the same place to have some confidence that something meaningful is happening. And so in Mitchell's study, he did 3 different mappings. One for staging, case versus control. So in his study, cases were B2, C, and D stage, and controls were B1 and A stage. He also did mapping for the LVIDDN ratio and for the LAAO ratio. And across all 3 of those mappings, we got signals at a few different places that appeared to be relatively consistent. And we actually did also found some functional changes. So in this chart here, what we're looking at, the higher the peak, the more significant, the more likely it is that it does something. If it goes above the red line, that's really, means there's something going on there. And so what we then need to do, each chunk of dots there is a chromosome. And so we know we can use technology to tell us exactly where that signal is coming from. And so we're looking for signals that there's a consistent signal in that place. And then we look there and we say, well, the data are telling us that something is happening here in relation to this measurement. What is here? And then so we look under that signal and say, are there any genes here that have been previously linked with heart disease? And if the answer is yes, then that gives us more confidence that we're finding something that might be important. And then we look in the DNA, every letter of the DNA, to see if there are any changes that might impact how those target genes function. And those then become our next set of genetic tests. So you can see here that there are probably another 4 or 5 potential genetic tests that might be used. Now, this is going to create its own problems, of course, and we're not quite there yet, but we've got some very good indicators here. At this stage, we are yet to do the validation for these. So remember, I was saying earlier, we don't believe it if we see it once, we've got to see it more than once. So the Swedes did that with the Dachshunds, and then we did it again with the Cavaliers. We need to do this in another set of Cavaliers to say, yes, these signals are not just an artifact of our local population, they actually are important for other Cavaliers too. And that research is not yet done. Alrighty, so now this is just a different way of looking at the same chart, but what it does is it piles the three different studies up on top of each other. So instead of doing a long line like we showed in the last one, we've got them in circles now. And what you can see though is that those light blue shaded areas depict where the same signals are being observed across all three of those studies. And so we have some hope that that we've got some consistent signals happening here, at least across these heart dimension and heart staging measurements for the breed. So with a bag of these tools in our hands, Nebel, and then Nebel won't show up in this because remember it was fixed. So that means that it's not mappable really. So we're not going to find Nebel using this approach. What we're going to find are genes that there are variable that are variable in the population, but when you deal the hand of cards, you can get a good hand or a bad hand, and we can then use genetic testing to make sure we get good hands more often than not. So that's the way we'd be going in the future. So the summary from the new research. We've got 4 new gene regions that have been identified. There are genes in 2 of the regions that have potential for genetic testing because we've identified actual protein coding changes in them. And the identified changes have strong potential to impact the cardiac health of the animals that are measured for them. But they're yet to be validated. Okay, so now it gets more complicated, right? Because Nebel's easy, we've just got one, we can deal with that. Soon we're going to be dealing with, if our research is successful, we're going to be dealing with several of these genes, plus other things that you're already dealing with that you're already going, oh, this is way too hard. So sorry?
I said, no, it's not. You have to start somewhere.
Oh, okay.
So it is hard, but you have to start somewhere.
So as the more gene targets are identified for more disorders, the breeding becomes more complicated. And you have to remember that no heart outcome is caused by one gene. So it's not going to be like PRA, where you get 2 PRA genes and you're going to be sick. Like for none of these do we know, none of these are like all or none. So all of these are just risk factors, not disorder causing genes per se. So what we want to do is to reduce those risk variants gradually. And you need to, until people start testing, you don't get a good feel for what the risk profiles of different regions are like. And sometimes you can go to, like, if you're in America, you might be able to come to Australia and get identify a bunch of dogs here that just don't have that risk gene. So that's a quick way to get rid of. One is to bring it in that you're always using that back crossing kind of approach that we mentioned previously. But all the time you need information, right? So, and until people begin testing, that information is not out there. So, for example, with the Nebel gene, we had no idea where the good dogs are until people start testing. And once people start testing, then the repositories of the good genes are revealed. So, at least in the early stages, testing is a good thing to do. Someone would like to ask a question. Barbara
page.
Barbara, do you want to type in the chat?
Questions will be at the end.
Okay.
What she's asking, Dr. Wade, is there an increased frequency of NEBL expression in humans with colorectal or lung NCS cancer? Please comment. That was that was one of her questions.
Oh, I'm sorry. That's outside of my expertise. I don't know. Why, there must be a reason why you think that would be the case. So far as I'm aware, I don't know. I don't know. I'm sorry.
Okay,
I'm not a physiologist, I'm a geneticist.
we can hold these questions till the end too.
Okay.
Okay.
All righty. So, sorry, I've got a lost puppy. Okay, so in general, there are mathematical solutions that can help, but these always have to be locally. Just a second, sorry. Sorry about that. I had a lost puppy. So the main thing to try and avoid is the biggest risk from having one of these holy grail genes is this popular-size syndrome. So this is something that should be avoided if possible. Try to avoid, as more people spread that gene out, the Nebel at least, probably start. It seems convenient to head for a dog that's homozygous, but I think I would go for a carrier. You're more likely to get more diversity in your breed. The last thing you want is to reduce your genetic diversity that you have, because it's only through having it's only through the availability of genetic diversity that we have access to these Nebel variants. If there was no genetic diversity, there would be no Nebel variants. So Nebel good variants. So it's important that people don't work all using the same sires. Try and spread the love a little bit. Now, I say that there are mathematical solutions, selection indices that can be used to help these. But the reality is, and I know this, is I think that they're unlikely to be available anytime soon for dog breeders. Maybe the UK, maybe Europe, probably not America, probably not Australia. We're just not organised enough, and there's just not that. The distances are big. The will in the registry organisations probably not there. They're costly. They require dedicated genetics people. And even in the UK, where they did have kennel club geneticists, they don't anymore. So it's unlikely, even though those things are probably the best way forward. I think it's unlikely that they'll be available in the near future. So in the end, you're just going to have to juggle it and just try and slowly increase the number of good genes and slowly decrease the number of bad genes, and just do it gradually. Don't go hard, because you have to balance it in all the other things that you're selecting for. So typically, what I would suggest to you is that I would try and fix the variants that have the most impact first. So the ones that have the most negative impact, those are the ones you should go for first. If there are disorder-causing variants, but they're relatively low frequency in your population, they might be good ones to target early too, because you can get rid of them straight away and then you don't have to test for them anymore. And if you're somewhere like Australia, and I can talk about this because we have this in our breed too, we have certain new diseases, genetic diseases that pop up from time to time overseas that do not exist in our local population. And so then I think the important thing is to not import the ones that you don't have already. So if there are risk variants that are identified, but they're not in your local population, then you don't have to worry about testing for those. You just have to ensure that all the imports don't bring them in so that you don't start having a problem later. Yeah, so it's a little bit hard to advise when these things are not there yet. But as rules of thumbs, I would say if something's nearly absent, get rid of it. Then you don't have to worry about it anymore. If something causes a lot of damage to the dog, and that's clearly been proven, get rid of those ones first, particularly the ones that affect the puppies when they go to their new homes. Yeah, and avoid bringing in problems that you don't already have in your local area or in your local breeding community. Right, so now there are also non-genomic approaches, and these have been used a little bit already, and some of you may be very familiar with them already. The one that first occurred to me was to use artificial breeding to help. So if you retain semen from promising younger dogs, but don't use it until the dog's older, then you'll know which group of dogs it was in, if it was in the group that the disease doesn't progress, or if it's in the group where the disease does progress. And if you use this semen from dogs with the longevity phenotype known, then very quickly you should be able to hone in on enriching the good genes in your population. Yeah, so using semen from dogs with known longevity phenotype would be the quick way. And doing that, you're capturing, it doesn't need a genetic test. You're capturing all those good genes by doing it that way. That's kind of like that's the proof is in the pudding. That dog's got the pudding. You're breeding from what you want to achieve. Yeah, so that's a non-genomic approach that you can use, artificial breeding. There are also existing breeding programs that are out there that I'm sure you know more about than I do, because I only learned about them last week. And that's, for example, the Cavalier Club in the UK has guidelines about screening for dogs, so that for your bitch, she should be a minimum of 2.5 years old with a clear heart and have parents with clear heart certificates at 5 years age or older, and same for the boys. And that they recommend not breeding under 5 unless they've got a clear heart certificate. So the other thing that they're doing that you can do, sorry, getting ahead of myself. So research has shown that kind of approach is actually successful at reducing MMVD over a longer term. So it takes time, but you will get there by using that approach. I guess nothing that we're talking about is quick fix. There's never going to be a quick fix. So, but this scientific paper, showed that they assessed dogs over that period of over a 10-year period, and showed that if the dogs were part of that a kind of breeding scheme that did heart screening, that they were much less likely to have heart disease than dogs that did not have that heart screening program. So therefore, they recommend that screening program. All right, so physical screening can be used as well as genetic screening. And the other option, this is another one that falls into the same category as selection index. Estimated breeding values are statistical constructs. They would require the archiving of echocardiography measurements in a central repository, and then they would require geneticists to regularly analyze those data to create scores for the dogs. And these kinds of approaches are used extensively in livestock breeding to increase animal production. They can be used for everything, really. They require the pedigree to be known, which is no problem for us. The main problem that dogs have with this kind of approach is that yes, this works great for livestock, but you've got to remember that in livestock, the way they breed is very, very different than the way we breed dogs. So in livestock, you've got 50 cows with one sire. You've got 1,000 cows in a herd. You've got bigger numbers. Like, we're more like dealing with the village goat situation where each of us has only a handful of breeding animals. You know, I've got one breeding bitch here and one dog. You know, like, the numbers are very different. Even the larger scale breeders among us, compared with livestock, we've got nothing. And so the way estimated breeding values work is they rely on the same genes being used in different places to estimate the local environment effect, and they rely on different genes being used in the same environment, so different size or dams being used in the same environment to estimate the genetic effect. And very rarely in our breeding programs does that happen, because we very rarely, you might have sires that are used in different environments. You very rarely, or I guess there are some situations, so you'd need to have different sires used in the same kennel environment in the same year, with puppies born at the same time, to get like a apples to apples comparison. And that kind of situation is extremely rare in dog breeding. And so while it is the state of the art for selective animal breeding, it's actually not probably very feasible for hobby breeder situations, at least not in Australia or in America, maybe in Europe, maybe in the UK. But again, you need access to that geneticist on a regular basis. And then you need a lot of training of people on how to best use them. And yeah, I think it's a long way off, to be honest, for dog breeders. So that's another approach that can be used. But I think for most people that I know, most breeders that I know, very likely it's not going to be a feasible approach for us. Okay, so just summarising, we're nearly at the end, you're nearly there. So there are lots of non-genomic approaches that are possible. Most of them require quite a lot of organisation to set up. And even then you have problems, because, I mean, we've got these kind of schemes for hip screening and have had. The guide dog agencies work okay with this kind of thing because they've got more of a production kind of setup. But for most hobby breeders, these large-scale breeding schemes are of limited, they're probably not going to be accessible in the short term. But one thing I used to always tell my students is that selection is incredibly powerful. You just have to not change your goal posts all the time. So the important thing about selection is you have to have a breeding goal and you have to be consistent in working towards that goal. And it's easier if you work towards that goal with other breeders who share your goal. And so selection, we know, is very powerful. It has given us all the different types of breeds of dogs that we see today. They all came from wolves. And so that's what selection can do. Selection can, that even when things seem fixed, they're not. Even though everybody says there's no diversity left in pedigreed dogs, that's not true. That's not true. And I can tell you a story about when I was at the Broad Institute and we were doing work with mice. In mice, they have these inbred lines of mice that were created in the 1950s. And these were deliberately inbred, brother to sister, mother to son, really, 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 really inbred. And so that they're all meant to be genetically identical. And so these were the mice that we used for the Mouse Genome Project. And one of the strains of mice was what would happen would be that the different laboratories would take these mice that were all meant to be the same and they'd take them off to their own laboratories and they'd breed them on. And so there was a study some years later, probably in the 1990s or noughties, where some scientists took these mice that were meant to be genetically the same as the other ones and they sequenced them. And they found that there was variation everywhere. So even when they had started out as being like identical, over time, nature creates new variation. It does that all the time. In average, you get 300 or 400 new variants every time a new egg is formed. So it's quite a lot of new variation that comes up. Now, the genetic testing, like the screening panels and stuff, they don't see it. And the reason they don't see it is because those variants that are used for genetic markers they have to be used in lots of breeds. And to be used in lots of breeds, it means they have to be very old. And so if they're very old, they can't see the new stuff. They can only see the stuff that's the same age as them. So there are, yeah. It's not as dire as everyone says, what I would say. But that's no excuse. That said, I am definitely not a proponent of line breeding. I don't line breed myself. In Australia, sometimes with a rarer breed, it's not possible to avoid relatives entirely, but on average, you'll lose one puppy from your litter with every 10% of inbreeding that you do. So I'm not a line breeder. I don't believe in it. I don't think there's any benefit in it. I don't believe in magic formulas. I think all of that stuff is untrue. I think a lot of people confuse line breeding with selection. So I am a proponent of selection. And so sometimes to select the things you want, you do choose animals from the same line. But the sake of line breeding, just to do it, there's no benefit in that, so far as I can tell. But selection, there is benefit in. Alrighty. So selection, yes. Line breeding, no. Popular size, no, no, no. Try and do things slowly, right? So try and do things slowly. Try and develop a community of like-minded breeders that you can work with. Alrighty, so our take-home messages, we're near the end. So just driving home, heart disease is not a simple trait and you can't repair it by selecting for one gene. So Nebel's not a magic bullet. The scientific evidence so far, however, does support that dogs with that Nebel3 wild-type variant will have a later onset of MMVD and possibly no clinical significance in their life. We have research that's ongoing to identify other genetic risk factors that might improve clinical outcomes for Cavaliers. And in the meantime, there are other non-genomic approaches that can be used. So heart screening, using semen from dogs that lived a long time, that can be used alongside gene testing to help you to move your population in the direction you would like to go at a more rapid pace. And so with that, I would like to very much thank the American Cavalier King Charles Spaniel Charitable Trust, the University of Sydney, who pays my salary, all 3 days a week of it, and Dog Breeding Science. So Dr. K.O. Castle from Dog Breeding Science was fabulous about going into her archives and bringing me the information that I requested so that I could share it with you in this presentation. So, and thank you all for coming along, and it's been lovely to have a chance to chat to you. And I'm happy to answer any questions.
Thank you.
Bye-bye.
Bye-bye now. Thank you.
Oh, thank you. You're welcome. Okay, everybody unmute. Are we still on?
Yeah.
Yeah.
Okay,
We gotta go.
we're still on. Wait, Steffi's gonna fix that for us.